At the Lockwood Lab, we've taken theory from traditional aquatic environmental DNA sampling methods and pioneered new methods in order to study terrestrial organisms. Just as aquatic life releases DNA into the environment, terrestrial organisms do the same in the form of exoskeletons, excrement, mucus, or skin cells. The DNA they leave behind is what we call environmental DNA, or eDNA. Having the ability to collect eDNA samples on land gives us the opportunity to efficiently assess the biodiversity of terrestrial ecosystems or assess the presence of a particular species of interest in that environment. Over the course of this video, we'd like to demonstrate one of the main methods we use to capture terrestrial eDNA off the surface of vegetation. This method is especially advantageous in agricultural settings. You'll see our method uses self-desiccating filter housings, but other commercially available and reusable filter housings can be used. Before we begin, we want to emphasize some standard practices that we follow and the importance of avoiding contamination when collecting samples. eDNA sampling is a very sensitive tool, so minute traces of contamination can cause false positive detections of an organism. To combat this, we thoroughly clean all of our sampling equipment with a 10% bleach solution, wear clean gloves, and change them often, between samples and whenever there is potential contamination occurring. We avoid touching any equipment that may come into contact with a sample, and we make use of negative control samples to detect any potential contamination. We typically collect field negatives using the same equipment used for sampling, but in the place of a field sample, we use deionized water. Ideally, a negative control is collected before beginning sampling at each field site, but minimally, field negative controls should be collected at the beginning and the end of each sampling day. As we go through our sampling process, you'll get a chance to see just how carefully we handle our sampling equipment. If interested, you can find a list of all the equipment we use for this particular method on the Rutgers eDNA Lab website. To begin sampling, you'll want to put on a fresh pair of gloves and set up your peristaltic pump and tubing. Be sure to keep the front end of your tubing clean throughout the sampling process to avoid any unwanted contamination. The back end of your tubing should be placed into a graduated cylinder in order to measure the amount of sample filtered, which can be an important variable for later analysis. Once set up, you can grab your sprayer and collection receptacle. We recommend handling all collection receptacles from the outside to avoid introducing any contamination from other surfaces in the field. With the collection bucket placed directly under your target surface, begin spraying the surface from approximately 1 foot away. Be sure that the area you are spraying does not exceed the diameter of your collection bucket, as you could potentially lose a portion of your sample. Continue moving your sprayer wand and collection bucket around until you've finished sampling your desired area of vegetation. Generally, it's good practice to standardize the area sprayed per sample. For example, you could spray along a 10-meter stretch for 1 minute per sample, maintaining a fairly consistent final volume. Some things to consider when sampling. You'll want to know where your target species feeds or spends most of its time, as this is likely where the most DNA will be deposited and available for detection. Surface eDNA isn't as well mixed across the landscape as it is in the case of aquatic eDNA, so targeting a specific portion of vegetation or a host plant species can mean the difference between a detection and non-detection of your target organism. When spraying, don't spend too much time on one section. We have found that DNA is easily dislodged from vegetation with the application of water, and moving around maximizes the likelihood you will gather some of your target species' DNA. Plant compounds can inhibit the downstream processes we use to detect organisms with eDNA, so try to minimize the amount of organic matter that ends up in your sample, such as dirt or detritus. Recent weather conditions should also be considered, as rain, in particular heavy rain, can wash away surface DNA. At this point, you can grab a clean filter. Typically, we pre-label the bag with the site name, date, and sample number. Prepare the filter housing with a fresh pair of gloves. Carefully attach the bottom of the filter housing to the pump tubing by using the filter bag to manipulate the filter. You'll then want to attach the snorkel provided to the bottom of the filter housing. Once your filter is prepped, insert your filter snorkel into the sample bag. The pump will begin to move your sample through the filter housing, capturing your eDNA field sample inside. Once all the water is filtered, you'll want to remove the snorkel from the bag and invert the housing. You can then remove the snorkel from the housing, slightly increase the speed of your pump, and let the pump continue running for another 30 seconds to allow your sample to dry. Detach the tubing from the back of the filter housing and place the housing in the labeled bag for storage. Be sure to remove as much air as possible before sealing the bag. The sample is now ready for transport in a cool, dark container, such as a cardboard box or cooler.
While our lab primarily uses self-desiccating filters for this method, you can use reusable filter housings. These assemblies should be thoroughly cleaned before use and assembled in a control setting whenever possible. Because they're not self-preserving, filters from reusable housings should be pulled as soon as possible and be kept cold or preserved in non-denatured ethanol to prevent degradation of the sample from occurring.
From the Rutgers eDNA lab and our partners, we hope you found this helpful. For more information on our sampling methods and eDNA in general, please visit our website using the QR code provided. Happy sampling!